So the title, or rather the theme of our presentation today is making genomic surveillance work. So, and essentially here, what we'll be looking at, or rather what we'll be discussing in this interactive session is what are the key decisions that we need to make when we are designing a genomic surveillance strategy. So just getting right into it. So the first decision, or rather the first key question that we should ask ourselves when designing a genomic surveillance strategy is asking ourselves, what situation are we really addressing? Because different situations require different surveillance strategies. So when we talk about the decisions or the situations, are we addressing an outbreak response, for example? Or are we performing a routine genomic surveillance? Or are we even performing pathogen genomic discovery? Because if we are looking for an outbreak response, for example, if that's what we are addressing, there are certain aspects that are really paramount to consider, such as turnaround time, for example, the speed and the rapid detection and containment measures. But if you're looking for, let's say you're addressing a routine genomic surveillance, aspects like depth, scalability, and cost efficiency tend to matter more, for example, other than the turnaround time. So knowing what is the objective that you're addressing is really key to guide you in developing a genomic surveillance strategy. There are several tools, of course, that we have to address genomic surveillance. We have culture, we have PCR. And at this point, I would like to quickly ask an interactive question that you can answer on what tool does your lab primarily use for pathogen surveillance or outbreak response? So you can scan the QR code on your screen, and then we can have a quick interactive session to understand the context around the lab that you are coming from. So maybe a minute or two. Okay, I hope you have had time to quickly scan the QR code. So let's quickly have a look at the responses.
On the screen for the responses.
Okay, I see there's some hands that have been raised. I hope you're in time to look at to scan the QR code.
The QR code is, I'm getting a different question. Oh, okay. I can see in your chat that you're getting a different question in the QR. Okay. So maybe display again the QR code. What question are you getting?
Which one is your favorite equipment?
Oh, I see. All right, we'll display again the QR code. Go to the PowerPoint. Okay. So maybe we can have a tally on the chat What tool does your lab primarily use for pathogen surveillance or outbreak response? Maybe you can type it in the chat. So let's use the chat box to answer the question. Okay. So I can see PCR, qPCR, NGS.
Okay. Yeah, PCR seems to be taking the day. Okay, NGS, PCR, NGS, PCR. Okay, fantastic. Okay, we can proceed. So these transitional tools, the PCR, culture, they have really served us well when we're doing our genomic surveillance. However, they are not without their limitations. For example, culture-based methods, they are gold standard in some of the assays for genomic surveillance. However, they tend to be time-consuming and they can take days to weeks. And you can really imagine if there's an outbreak situation where you need to respond quickly and present your findings, it can be a bit frustrating to use such kind of an approach to address an outbreak situation timely. PCR-based methods, well, we well know they are rapid and they are very sensitive. But we have situations where the primers can fail, especially if mutations tend to occur in your primer binding sites. So what we're really saying here is these tools have really served us well, but in the current landscape of genomic challenges that we are facing, for example, during the recent pandemic that we had with the SARS-CoV-2, a timely solution or two really comes in handy for us to address, for example, an outbreak response. So what makes NGS so powerful as a genomic surveillance tool? There are several benefits that builds the case for NGS utilization. Of course, using, for example, a shotgun metagenomics approach, you can do unbiased detection of known or unknown or even emerging pathogens. You can get very high and deep genomic resolution when using NGS, especially because of the aspects such as depth and coverage that this technology provides. You can do your transmission tracking. You can detect multiple pathogens. For example, when you use certain specific panels that target multiple pathogens in a single assay. You can do your variant monitoring. Of course, the aspect of high throughput, especially if you're doing a large scale population surveillance, is a added advantage that you get with utilization of NGS. Rapid outbreak investigation, antimicrobial profiling. I mean, we can build a very endless case for what makes NGS so powerful and why we really need to utilize it in our genomic, or rather why we need to highly consider it in our genomic surveillance strategy. With all these advantages that we have for NGS, our next interactive question is, we'd like to know what is our biggest bottleneck that affects us, or rather that makes us not adopt NGS. So please take your time and quickly scan the QR code. Or rather, we can use the chat. I think the chat is working faster. Yeah, we'd like to get some context on what really is the biggest bottleneck. Maybe a minute on that. Funding, okay. We have a funding response. Cost, okay. Skills training, okay. Supply chain and reagents. And if there's any other bottleneck that we've not highlighted here, please feel free to also include it in the chat. It will enable us to have more context on what are the real bottlenecks that affect us adopting NGS.
Lack of awareness.
Lack of awareness. Yeah, that's interesting one. Charles. Service maintenance sustainability. Okay. Okay, maintenance.
Takes too long to order and get reagents.
Okay, okay.
Interesting. Okay, oh, data storage.
Okay, data storage. Okay. Expensive. Wow. Okay. Okay, maybe at this point, okay, thank you very much for those who have quickly highlighted the bottlenecks. Maybe at this point, we would like rather to know if there's any researcher in the chat or in this call that has been able to maybe mitigate a particular bottleneck, let's say funding, and maybe can quickly tell us how he or she was able to mitigate around a particular bottleneck in his or her scenario.
Just put the hand up.
Yeah, sure. Please put your hand up and you can quickly share with us what strategy you adopted to solve that bottleneck that can be useful to the rest of us. Maybe as we wait for that, maybe funding, what we can highlight on that is collaborations, I think, are key to navigate the issue of funding. For example, if you don't have funding for CapEx, but you're funding for reagents and consumables, you can always look for and to collaborate or partner with laboratories that already have the instrumentation. So it can be a strategy to navigate around a bottleneck like funding, for example. Skills of training, I think, okay, from an industry point of view, this is something that we are taking seriously in providing capacity building to be able to adopt NGS. Okay. Yeah, so, but at the end of the session, you can have a discussion around this, how we can help each other solve some of these different bottlenecks. Okay, so let's proceed. When we talk about NGS workflow and making decisions for when you're designing a genomic surveillance, first of all, it is important to look at the workflow in entirety. That is, what exactly is required for you to learn an NGS lab, for example. The aspect like sampling, of course, that you need to consider what samples are you working with, how do you source them, for example. What are the extraction strategies for you to get your nucleic material for downstream processing? That's for your library prep and sequencing. And of course, your data analysis. So when we work in the lab, when we execute the workflows, of course, we tend to think in a chronological manner, like from sampling, we then perform our extraction, we then perform our library prep, sequencing and data analysis. However, when we want to do strategic decision making in terms of what's the objective of what you're trying to address, the most important output that we should always have in mind is the data. So we tend to say that the laboratory is a factory, but the data is a product. So we should always have the design of the data in our mind when we are making a strategic decision. So what do we mean by this? For example, if your key objective is to address an outbreak response, one of the key decisions that we should be making around is how quickly are we able, for example, to report our findings in a timely fashions. So the idea here is always consider your data needs first, always design your workflow backwards, when you're making your decision making, because that comes a lot in handy in executing, or rather in presenting the findings of your objective. Because we tend to have scenarios where we have these sequencing platforms, you've generated your data, and then it takes quite a while to put that data beyond the lab, to publish it out there and inform public policy and public health decisions. And the key questions that we should address, of course, should be guided by the objective that we are trying to aim at. So when you look at, for example, if you're an outbreak response objective, aspects like turnaround time should really guide our decision-making process. Think aspects like high confidence variant detection, for example, whether we have standardized data analysis pipelines, whether we have quality control threshold to give us confidence in the data that we are reporting, and of course, the aspect of scalability during such events and actionable reporting will be some of the key factors that will really guide us in developing a very robust outbreak response genomic strategy. If we are aiming to address an aspect like routine genomic surveillance, not to say that turnaround time will not be critical, but there are other aspects that tend to be to matter more. For example, how the throughput of what we are processing, cost effectiveness of data generation, for example, whether we have a cloud-based infrastructure or standardized data formats. Because here you're doing more of long-term monitoring of a situation, and depth and scalability will really matter in this aspect. If our key decision or the aim that we aim to address is pathogen discovery, for example, aspect like whether we have large data volumes, for example, if you're doing a shotgun metagenomics approach, will really come in handy in your decision-making process. Whether you need advanced bioinformatic tools to be able to, for example, discover those variants with confidence. Aspect like deep sequencing coverage will really be critical in this aspect of decision making, or in this particular objective. So just to again highlight, depending on the goal, or rather the objective of what we are really aiming for, and this should be guided from the data that we are aiming to present or to analyze at the end of the day, will really impact what kind of approach or what kind of strategy we should quickly aim for. At this point, we'll have another quick interactive question. Again, feel free to respond in the chat. So in this question, you're responding to an outbreak which is spreading rapidly, and your PCR tests are all negative. So, and you're considering to utilize NGS. What are some of the key considerations we should make? Is it selecting the fastest throughput sequencer, for example, or choosing the most efficient extraction kit, or defining our bioinformatic pipeline and reporting requirement, or is it choosing the best library prep kit? Again, feel free to quickly drop your response on the chat. Selecting the fastest throughput sequencer, okay. So remember, this is an outbreak situation. You want to quickly report your findings, so your reports. Just a hint, there can be more than one correct answer. So all those apply, okay.
Best short library prep kit.
Choosing the best library prep kit, or the above, okay.
Sequencing deep to increase sensitivity.
Okay, sequencing deeper to increase sensitivity.
why are they important?
Of course, there can be some other key considerations. Okay, I think we have enough responses. So for sure, all of these considerations are important for a rapid outbreak response. You need a fast sequencer so that you report your findings in time. Of course, you need an efficient extraction kit because you don't want to spend all the time extracting and possibly exhausting your sample while at it. Of course, having a very defined informatic pipeline will come very much in handy when your data is generated, because it will help you to report your findings very quickly. And of course, choosing the best library prep kit is very, very important. So there can be other considerations as well. The key thing here is to understand that every aspect of NGS workflow can have an impact when you're trying to address a particular genomic surveillance objective. Okay, so quickly going on to the next part. So we've seen that when we are designing our genomic strategy, we need to think it from the back forward, starting with data decisions. What are the key data decisions that we should be making so that we are able to address our objectives well? So this trend is more of a thought process. Of course, there are more other key factors that we can consider. And again, so we start with the goal in mind. What are we really addressing here? Is it a rapid outbreak detection? Is it routine surveillance? Is it pathogen discovery? In our data tool that we are using, is it targeting whatever that the data or the insights, whether is it from viruses? Do we want insights about bacteria from this particular objective? And what is the decision strategy that is guiding the data that is being generated? Is it a targeted approach analysis? Is it shotgun metagenomics, for example? If it is an outbreak response, for example, is my strategy incorporating the fact that I want maybe quick pathogen ID information, or maybe viral characterization if I'm looking at viruses, or maybe AMR information or any other epidemiological insights? Of course, the other key decision is the speed of analysis. How quick is the tool that I'm utilizing to do my data analysis? If, for example, if you only have available data applications, would it make sense to start developing pipeline from scratch or utilizing what is available? So these are kind of decisions to make. Compute infrastructure, what kind of infrastructure should we consider? Should we consider cloud platform? Should we consider local workstation? Should we consider high performance computing infrastructure? So we need to have this in mind, whether we have the resources ready to address our objectives. Of course, the data output, for example, for outbreak surveillance, you want for public health laboratory, for example, you want processed summary outputs to quickly disseminate your data. Are you interested in getting bioinformatic outputs like FASTQ faster for you maybe to do other custom analysis to address maybe specific objectives of your study, of your research project? Of course, decisions around whether you're getting a public health report, whether you're getting lineage information, come very much in handy when you're making your data decisions. Okay, when we come now to the library prep and sequencing decisions, again, you'll notice that aligning each aspect of your NGS workflow with your goal makes a lot of sense. It helps you to it helps us faithful to the specific objective that we want to address. Again, is it viruses that we are aiming for? Is it bacteria? Is it a broader scope kind of approach that we want? Are we going for a targeted shotgun? Are we looking at a single pathogen or multiple pathogens? This will guide you on what, for example, library prep kit you utilize in this case. What genomic resolution do we want? Do we want just quick ID information of what is present in our sample, for example? Do we want full genome characterization information? Do we want variant analysis? This is really key, again, when deciding which library prep kit to use. Depth, the level of depth that you want, and of course, the level of throughput, how much of the library prep that you're using, how many samples can it handle. So these guide us a lot in making the right decisions in this aspect of the workflow. Quickly, when you look at the pathogen scope decisions that we need to make, again, the objective of our study is key to keep in mind. What sample type are we dealing with? What's the collection method? Are we storing our samples in the right medium, for example, to ensure that the integrity of our nucleic material is not affected downstream? What is the storing strategy? Are we storing our samples in short-term resource, whether it's in +4 or -20 or -80? Are we employing a manual automated way of extracting our nucleic material? Are we interested in DNA or RNA or in the total nucleic acid? Again, these decisions are very important. We need also to consider what are the freeze dose threshold that we can go for example. And of course, importantly, the QC of your nucleic material can't be overemphasized enough. It's very important because whichever approach that we will use, there'll be a threshold in terms of what is the optimum QC yield that we require for our downstream processing. So how do we apply NGS in practice from an Illumina workflow perspective? Okay, when we start making decisions about data analysis solutions, depending on the objective that you're addressing, one of the key questions to ask ourselves is, let's say it's an outbreak situation and you want to really report your findings quickly. What are the available data analysis applications that are available? For example, are there applications that are ready, like, for example, what Illumina Base Space provides for you? So finding whether there are ready applications that can be utilized makes a lot of logical sense to go and try them. Of course, again, depending on the objective, the data analysis solution should be guided by what really you are aiming for. But in a case where you're addressing an outbreak response, for instance, trying to see what is already readily available goes a long way in making your outbreak response timely. Just quickly highlight, there are quite a number of data analysis applications that are provided by Illumina from the Base Space resource, where they have curated different pipelines depending on the applications that you're using, and they're worth looking at. For example, for infectious disease, if you're utilizing, for example, a panel like the hybrid enrichment panels, some of the data analysis applications are free to use, and they can really accelerate your report finding if, for example, you're addressing an outbreak situation. Of course, there are different levels of analysis that are provided by Illumina, again, helping you address whatever you really are looking for. We have tertiary analysis. This is more a deeper analysis solution where you can get more insightful information beyond what is being provided from a secondary analysis point of view. So again, it makes a lot of sense to see what is available and whether it addresses your specific objective before exploring other different tools. There's nothing wrong, of course, in exploring other different tools. And at this point, we would like to know what's your main limitation, for example, when it comes to utilizing cloud data analysis platforms. Is it limited internet or bandwidth? Again, feel free to drop a response on the chat. Is it lack of training or expertise? Is it has to do with maybe cost or subscription issues? Is it data security or compliance? Or possibly is it because we don't know where we can find some of these cloud solutions? So again, maybe a minute or two.
So we're already getting the answers. So there's lack of training for the free ones. There's data security and compliance. We have the lack of training, expertise. There's cost issues. I think data security is coming up again. Cost
Okay.
of subscription. Yeah.
The cost of subscription, data security, lack of training. Okay.
So internet is not a problem in Kenya.
Okay,
Or-
that's good to hear. Cost and data security. So, okay, we can agree internet and bandwidth is not an issue in our country, so that's impressive.
Okay. Right. So
I guess most of the people know where to find cloud solutions, which is also impressive to know. Okay, any other response? Subscription issues. Okay.
compliance and security concerns.
Compliance and security concerns. Okay. All right. So what are the key decisions that we need to make when it comes to addressing our library prep decisions? So the first thing that we need to understand is what are the different approaches that are there for us to do our library prep, okay? Are we interested, is the objective that we are aiming for going to be addressed by whole genome sequencing approach, for example? So by whole genome sequencing, of course, we mean we want to do full genome characterizations of our pathogens and identify, for example, potential causative variants for deeper studies. Is an amplicon approach going to address our situation, for example? For example, you remember during the COVID pandemic, the amplicon approach was readily utilized when doing the SARS-CoV-2 surveillance. And one of the key reasons was the cost effectiveness and also the speed of this particular approach. So if speed, cost effectiveness is an aspect that you highly consider, for example, an amplicon approach can be used in an instance, for example, instead of using an whole genome sequencing approach. So again, it's important to understand what we are aiming for. An hybrid capture enrichment approach. This is where you're using different probes in a panel format to target a lot of things, a lot of pathogens, for example. So we have hybrid capture enrichment panels, for example, that are curated to target quite a number of things. For example, Illumina provides you with the virus surveillance panel, which supports if you are looking at viruses. We have the respiratory pathogen panel, for example, which looks at quite a number of pathogens, over bacteria, viruses, and so on and so forth. So if an hybrid capture enrichment approach, for example, in an outbreak scenario, and you want to have an idea of what is really circulating, an hybrid capture enrichment approach would make quite a lot of sense in quickly fishing out what are the pathogens that could be in my sample, for example. Of course, we have a metagenomics or metatranscriptomic approach. Metagenomics is where we want to see what pathogens might be present in the sample. Metatranscriptomics is where we want to study gene expression, understanding maybe what are the microbial genes that are active in a particular sample. So as you can see on this slide, we have different solutions depending on the different objectives that you want to address. So if it's an whole genome sequencing approach, you have the Illumina DNA prep, for example. We have the Illumina microbial amplicon prep, which basically is a very versatile library solution where you can replace the primers of interest and address whatever, for example, virus that you're looking at. So for example, if you're looking at chikungunya, if you're looking at maybe rhinovirus, you can switch the primers because the backbone of this particular library prep can support quite a big or a broad spectrum of viruses of interest that you're looking at. When it comes to hybrid captures, I've mentioned we have different solutions like the Illumina RNA prep, which I'll quickly highlight. So for example, this particular panel can come very much in handy when you want to identify what is really in your sample. So it can identify more than 282 pathogens that are associated with lower respiratory tract infections, over 52 fungi, over 40 viruses, over 180 bacteria, for example. In addition to that, you get antimicrobial resistance profile information. So if your particular objective is to understand really what is really going on, maybe circulating it can be in an outbreak situation, or maybe just in a routine surveillance system, this is a panel that can quickly come in handy to help you address that particular objective. And another key advantage that comes with this particular panel is the data analysis out of this data is freely powered on the Illumina base cloud data analysis solution. So really, it helps you to quickly address a situation like an outbreak. Furthermore, if you're looking into maybe addressing more deeper maybe objectives, there are faster files that are generated from these pipelines that can be used for downstream third-party analysis. So it's really a win-win situation, whether you're for whichever particular objective that you're aiming to address. If your aim is to look at the viruses that are circulating, for example, we have the viral surveillance panel again, which covers quite a broad spectrum of viruses, over 200 viruses, and again, the data analysis for the data generated for this particular approach is readily available on the Illumina's cloud solution. So let's address now the sequencing decisions that we need to make. So let's say we've settled it is an outbreak, we don't know what's circulating, we don't know whether it's a bacteria, whether it's a virus, whether it's a fungi that's wreaking havoc. And we decided to select the RPIP hybrid enrichment panel to do our maybe respiratory genomic surveillance or outbreak response. The key question that can come here is, what instrument maybe do we need to select to be able to address this? So one of the key things that Illumina does very strongly is scalability when it comes to these solutions. As you can see on this screen, there's quite an array of options on what, for example, flow cell configurations that you can go for. In terms of instrumentation, there are guiding points in terms of specs on how much, for example, a run can be performed on a particular instrument. Let's say we are doing a 300 cycle run on any of the flow cells that I highlighted. If you're performing a run on the MySec High 100, you can see that you can address a situation very quickly if it's an outbreak situation, because within 7 hours, you already have your data generated. And something that I should mention at this point is all these Illumina sequencing instrumentations, they have onboard data analysis on them. The key flagship sequences that I will highlight there later, they have onboard data analysis on them. So you are not like transferring your data. You can do your data analysis onboard the instrument and get really insightful data. Of course, there's a cloud option, but just to quickly highlight, the integrated data analysis option can come very in handy in addressing your particular situation as well. If throughput is your concern and you want to sequence as many samples as possible, bigger instrumentation like the NextSec 1000 platform can quickly address that kind of a question. For example, if you want to process as high as 200 samples at a go, or even 384 samples at a go, so there's this flow cell configuration that you can go for. So again, as you notice here, the data that you want guides most of your downstream decisions. That's why it's very important to have what is the data expectation or anticipation that you want to be able to know which is the best strategy to go for. Again, at this point, let's have a quick interactive question. So you want to perform genomic surveillance of respiratory pathogens, maybe bacteria, viruses, fungi in a sample. So which approach makes sense for you? Is it an whole genome sequencing approach, for example? Is it an apricot sequencing approach? Is it a hybrid capture enrichment approach, where you're targeting multiple pathogens at once? is it a metatranscriptomics approach? So again, feel free to quickly drop your response on the chat. Okay, hybrid capture. Amplicon and hybrid capture, okay. Or can apply depending on capacity, okay.
And hybrid.
Amplicon and hybrid capture, okay.
Whole genome sequencing.
Whole genome sequencing.
And which is the right one?
Okay. So yeah, we've gotten quite a number of responses. So there's whole genome sequencing, there's an amplicon sequencing approach, there's hybrid capture, or can apply depending on the capacity. Okay, it depends on the questions we want to address. Okay. Yeah. All right. Yeah, this is quite interesting. So again, let's see. You want to perform genomic surveillance of bacteria, viruses, fungi in a sample. So which approach makes sense? Yeah, well, you can do whole genome sequencing using a shotgun metagenomics approach. The caveat here may be you'll need a lot of data, for example, to be able to address that. For example, if you're using a shotgun metagenomics approach using the Illumina DNA prep solution, you'll require about 10 millilins per sample to be able to get all these pathogens. So capacity here will be important. If the capacity is there, as one of our responders has said, that approach we can go for that particular approach. Amplicon sequencing, well, it's cost effective if you have the primers, but it can be cumbersome if you're looking at all these things, bacteria, viruses, and fungi. So you can imagine how many number of primers you need to design for all these. So in terms of making sense, in some scenarios, this will not make a lot of sense if you're looking at so many multiple things at once. Hybrid capture enrichment. So for this particular question, hybrid capture enrichment makes a great deal of sense because you have a panel, for example, like the respiratory pathogen ID panel, which can be able to capture all these pathogens at once, and therefore can make a lot of sense in this kind of scenario. Metatranscriptomics, not really, because unless your key goal is addressing gene expression, yeah. So this will not make a lot of sense in this kind of a scenario. Maybe another quick question. For a rapid outbreak response, where you need to inform containment, which is our most preferable sequencer? Is it the MySeq I100, the new platform from Illumina? Is it the NextSeq 1000, 2000? Is it the NovaSeq X? Again, feel free to drop your responses in the chat. MySeq I100, okay. MySeq I100. Awesome. Okay, we will, for the sake of time, we'll quickly go. So indeed, the MySeq I100 will make a lot of sense. It's one of the upgrades from Illumina, the latest instrumentation from Illumina, where you can do your sequencing for as low as 4 hours and up to 15 hours, depending on the flow cell configuration. And it can come in very much in handy if you're addressing an outbreak response because of the speed it takes to do data analysis, and also for the fact that there's onboard data analysis solutions that on the instrument. So you get a very complete solution if you're addressing such kind of a goal. This is quickly highlight the three key flagship sequencers that we have from Illumina, of course, from a scalability, flexibility point of view, and also depending on what exactly that you are looking for. So we have the MySec I100, which comes in 2 versions. We have the NextSec 1000 and 2000, again, comes in 2 different versions, addressing the aspect of scalability and throughput. And also we have the NovaSeq X, especially if you're looking at production scale kind of sequencing. So what are the quick takeaways from our webinar today? So a quick recap. So we've seen that for different situations, we require different surveillance strategies. And also we've seen that when we are performing a strategic decision-making process, the most important output that should be in the back of our mind is the data. The data guides us on what downstream decisions to go for. We've seen that there are multiple sequencing strategies we can go for to address different objectives. Again, we've seen that there are integrated data analysis ecosystems like the Illumina Dragon applications, which are available on board and also on the cloud and can be leveraged to address a particular situation. And we've seen that the aspect of scalability and flexibility that is provided for you to address different goals objectives. So I will end at this point and mention that the most effective genomic surveillance programs usually prioritize flexibility, scalability, and integrated data analysis, which Illumina does very well in supporting. At this point, I'll hand it over to Dr. Cecilia to do a quick conclusion. And also, I don't know, do we have time for a quick discussion?
All right, so this would be our time for questions and maybe a quick discussion. I think we have about 8 minutes. In case you have a question, maybe you can raise your hand and then your mic will be unmuted, or you can ask a question in the chat and we will be able to address it. So I think Peter has done a very good summary, and thank you, Joho. You said nice presentation. I'm still using your presentation. So I think we have had a very good summary. Essentially, for me, what we have captured today is how do we make the decision on adopting genomics surveillance as a tool for what we do in the lab, be it outbreak response, be it routine surveillance, be it pathogen discovery, and maybe to highlight from the questions that we have answered, because this was a very good point of information. I think a lot of us are doing our surveillance using PCR and qPCR. And as Peter has highlighted, these have been great tools and very informative tools. But how do we make the decision to now progress towards genomic surveillance to gain more insights and more data? So what are the key considerations? And there've been maybe data, what instrument would be best, what do we routinely do? Is it surveillance? Is it outbreak? Is it discovery? And maybe one of the questions we did not address a lot was on the cloud utility. I could see a lot of comments about the cost and the training and the expertise. So going forward, I think one of the things that we would love to do as part of our conversations is maybe also prioritize the trainings around data analysis and the tools that are available for Illumina. So for being part of this webinar, I will share with you a link that will give you a very good guide. The Gain Greater Insights into Microbial Mysteries is a guide that will maybe summarize and contextualize what we have talked about today. And maybe you can look at the different solutions that are available within the decision matrix that you need to make when thinking about adopting genomics as part of your work. So there's a question here, does Illumina have or is it working on any field condition level sequencing? Maybe something that has less trained staff and controlled lab environment. So as Peter thinks about that, let me just highlight, he will address that, what we have next in store. Is the next slide. So following this talk, we hope that you have gained some insights around what questions to ask yourself as you're considering maybe to scale up, or if you have scaled up, what are the other considerations that you make around a project? So what we're looking at next is within the next few months, we will have a detailed virtual workflow training on a selected library workflow, because it is also important to look at what does it entail to run a workflow from beginning to end, where you have the sample all the way to data. And that will be followed by a hands-on library prep workflow for selected individuals that will come from that second training. So this will be announced, and I hope you'll get the opportunity to apply and maybe join the lab team in doing the end-to-end workflow. I think some of you are already doing these workflows routinely and are excellent at it, but we'd love the opportunity to also introduce some of you to the end-to-end workflow. Then that will be followed with the data analysis training on the drug and microbial enrichment plus tool. I've seen some people have said the training and maybe the exposure to these tools might be a challenge. So we're looking forward to that in the future. And as Peter addresses the question that was raised, I'm just gonna leave our contacts there in case you have any questions or you'd like to follow up with us. Those are our contacts, Peter and I. We are both genomic application specialists here at FNDS, and we'd love to engage with you and maybe answer your questions and follow up on anything that could have arisen from this talk for you. So Peter, the question that you need to address is, does Illumina have any?
Does Illumina have working or are working on any field conditions level sequencing? So I presume the question here is whether we are looking for a portable solution. Unfortunately, the the product team doesn't review this information for us. I can't really guarantee whether they're working on something on this level, but you never know what's in store for the future. Something that I believe they're aware of. So let's keep this space open and see whether it's in the plans. Okay.
So another question is, does Illumina use proprietary data formats within BaseSpace that might not be universally usable? So no, Illumina uses standardized data formats. So the FASTQ, VCF files, the data that you upload and analyze within the cloud or even your BaseSpace account is the standard universally available data formats. So yeah, it makes it very flexible and accessible. And in terms of less lab control lab environments, I'd say the current MySekai 100 is actually quite friendly in terms of usability and mobility. And the fact that it uses room temperature storage, that the reagents are okay at ambient temperature, means it's more versatile within a collaborative network, because you can use it across the network in different lab spaces. It might not be pocket size, but it is friendly for mobility. That's my take on that.
Absolutely, yeah. So the MySekai 100, they say it was designed for Africa to address aspects like infectious disease surveillance. So it's such a handy tool to try. So there's another question: does Illumina have access to all data shared on BaseSpace? No, and a big no. We don't have access to your data. So the BaseSpace ecosystem is an ISO certified ecosystem where they have taken a lot of considerations to make sure aspects like data privacy are taken very seriously. And just to mention, Illumina is an IPO, it's a listed company in the New York Stock Exchange. And aspect like data privacy, of course, if they leak, can really be detrimental to a company like Illumina. So that gives you another layer of confidence to know that we really don't access your data at all. So you work in a sort of containerized account system where only you have access to your data, But of course, you have the privilege to share your data with the collaborators.
So yeah, data security is a very serious very serious consideration for Illumina. So you are the only one who has control of who accesses your data collaboratively or individually. Once you create account, the access controls sit with you.
Yeah, so we really don't access your data at all. Yeah.
All right. So it's 12. We like to be on time. And thank you so much for making time to attend this session. And we look forward to more interactions. Next time we'll have a bigger window for discussion. And look out for the announcement for the next upcoming virtual training on library prep workflow that will be leading to the hands-on. We will be sharing with you the link to access the guide in the same channel that we invited. I wish we could paste it. We can paste it in the chat so that before you leave, you can have you can download it from online. So before you check out of the meeting, kindly give us just 1 minute to paste that link onto your chat. And thank you very much. Looking forward to future engagements with you. Asante.
Thank you very much and have a good day ahead.
Okay. Perfect. So there you go. That is a very useful guide for you. And have a lovely afternoon.
Thank you very much.